Picture this. In 2012, Russian archaeologists working in a Siberian cave found a bone fragment so small it could fit on your fingertip. Just another piece of ancient debris among thousands of others. But this tiny fragment would shatter everything we thought we knew about human evolution. The bone belonged to a 13-year-old girl who lived 90,000 years ago. When scientists sequenced her DNA, they discovered something that shouldn't have been possible. Her mother was a Neanderthal. Her father was a Denisovan. She was a first-generation hybrid between two different human species. They called her Denny. She wasn't supposed to exist. But here's what makes this discovery earth-shattering. If your ancestors left Africa, you carry the genetic legacy of encounters just like the one that created Denny. You're not pure Homo sapiens. You're a mixture of different human species that met, mated, and passed their genes down through thousands of generations. These ancient genetic gifts both saved and cursed us. The same DNA that helped our ancestors survive Ice Age winters, fight off deadly pathogens, and adapt to crushing altitudes now drives modern diseases. We carry within us a genetic time bomb, variants that were lifesavers 50,000 years ago, but have become silent killers in our modern world. This is the story of humanity's most consequential bargain, written in the language of our genes. To understand how encounters like Denny's became possible, we need to go back 50,000 years. The world was colder then, locked in an ice age. Sea levels were lower. You could walk from Britain to Europe, from Siberia to Alaska, and humans weren't alone. In the Levant, that crossroads between Africa, Europe, and Asia, 3 different human species were about to collide. Modern humans were pushing out of Africa. Neanderthals had dominated Europe and Western Asia for 300,000 years, and somewhere in the vast expanses of Asia, the mysterious Denisovans had carved out their own territories. Forget everything you've seen in cartoons about Neanderthals. They weren't the brutish cavemen of popular imagination. Archaeological evidence paints a completely different picture. They had brains as large as ours, sometimes larger. They made sophisticated tools using techniques that required planning multiple steps ahead. They controlled fire, built shelters, and crafted clothes from animal hides. At Iraq's Shanidar cave, researchers uncovered Neanderthal remains with pollen from flowers scattered around the bones. The discovery sparked debate. Was this intentional flower burial, or simply natural pollen drift? The deliberate positioning of the bodies pointed toward ritualistic care for the deceased. In Spain, cave paintings dating back over 64,000 years show hand stencils and geometric shapes painted with red ochre. This was before modern humans reached Europe. Neanderthals made art. They even made jewelry, eagle talon pendants, perforated shells, carved bone ornaments. At one Spanish site, Neanderthals built sleeping areas with grass beds that they kept clean by burning them periodically. They used toothpicks made from bone. They processed plant foods, cooking starchy tubers. Analysis of dental plaque reveals the use of medicinal plants, including chamomile and yarrow. One individual had taken a primitive form of aspirin, poplar bark containing salicylic acid, while suffering from a dental abscess. Meanwhile, the Denisovans occupied territories stretching from Siberian caves to the Tibetan plateau. A Denisovan jawbone found at an elevation of 10,000 feet dates to 160,000 years ago. They lived at altitudes that would challenge modern humans without genetic adaptations. When these groups finally met around 47,000 years ago, something remarkable happened. Recent genomic analysis has pinpointed this encounter with unprecedented precision. The main pulse of interbreeding lasted roughly 7,000 years, from 50,500 to 43,500 years ago. The location was probably the Middle East, where Africa meets Eurasia. This was the bottleneck where expanding modern humans first encountered their archaic cousins in significant numbers. All non-African populations today share the same basic Neanderthal heritage from this period, suggesting a small founding population that interbred and then expanded across the globe. But here's where the story gets strange. While we carry substantial amounts of Neanderthal nuclear DNA, 2 specific genetic lineages have vanished completely. No modern human has ever been found carrying Neanderthal Y chromosome DNA or mitochondrial DNA. The Y chromosome passes from father to son, Mitochondrial DNA passes from mother to child. Their complete absence is statistically impossible by chance alone. The most likely explanation involves biological incompatibility. After 500,000 years of evolutionary divergence, Neanderthals and modern humans were on the verge of becoming separate species. The Neanderthal Y chromosome contained immune-related genes that differed from ours. Pregnant modern human mothers might have recognized male fetuses carrying these foreign proteins and mounted immune attacks, leading to higher miscarriage rates for hybrid males. Similarly, Neanderthal mitochondrial DNA may not have functioned optimally with modern human nuclear genes, creating subtle disadvantages that accumulated over generations. The evidence suggests these weren't rare, isolated encounters. When different human groups met, they saw something familiar enough to mate and produce fertile offspring. They recognized each other as human, despite their differences. These ancient encounters weren't just romantic meetings. They were evolutionary game changers. When our ancestors interbred with Neanderthals and Denisovans, they weren't just exchanging genes. They were downloading pre-tested survival software. Think about it. Instead of waiting thousands of generations for random mutations to slowly adapt to new environments, modern humans acquired genetic solutions that had already been refined over hundreds of thousands of years. This was evolution on fast forward. Archaic humans had spent millennia adapting to Eurasian climates, pathogens, and altitudes. Through interbreeding, we inherited their evolutionary shortcuts. The most dramatic example comes from the roof of the world. The Tibetan Plateau sits over 13,000 feet above sea level, where the air contains 40% less oxygen than at sea level. For most humans, this creates a deadly problem. Their bodies compensate by producing massive amounts of red blood cells to capture more oxygen. But this thickens the blood dangerously, leading to strokes, heart attacks, and life-threatening complications during pregnancy. Tibetans don't have this problem. Their blood stays thin. Their hemoglobin levels remain low. They've adapted to altitude in a completely different way. For decades, scientists couldn't understand how they evolved this superpower so quickly. The Tibetan Plateau was only populated about 25,000 years ago, not enough time for such complex adaptations to evolve from scratch. The answer came when researchers sequenced the EPAS1 gene in Tibetans. This gene regulates the body's response to low oxygen. The Tibetan version is radically different from other modern humans. It's so different that scientists couldn't find its origin in any living population. Until they checked the Denisovan genome. There it was, the exact same variant. Tibetans hadn't evolved their high-altitude superpower independently. They inherited it. Somewhere in Asia, as modern humans spread toward Tibet, they encountered Denisovans who had already adapted to thin air. Through interbreeding, they acquired a genetic solution that had been refined over hundreds of thousands of years. Today, this Denisovan gene variant occurs in nearly 90% of Tibetans, but only 1 to 2% of lowland Chinese populations. Natural selection rapidly increased its frequency once Tibetans moved to high altitude, transforming a neutral genetic variant into a life-saving adaptation. But altitude wasn't the only challenge. As modern humans entered Eurasia, they walked into a new world of diseases. The bacteria, fungi, and viruses of Europe and Asia were completely foreign to immune systems tuned for African pathogens. This is where Neanderthal genes became lifesavers. Neanderthals had lived in Eurasia for over 200,000 years. Their immune systems were battle-tested against local pathogens. 3 toll-like receptor genes, TLR1, TLR6, and TLR10, show unusually high Neanderthal ancestry in modern Europeans and Asians. These genes detect invading microbes and trigger rapid immune responses. When our ancestors encountered unfamiliar diseases, Neanderthal immune genes provided immediate protection. It's like having antivirus software pre-installed against threats you've never seen before. The genes were so beneficial that natural selection rapidly increased their frequency in populations entering pathogen-rich environments. Climate presented another challenge. Moving from tropical Africa to cold, low UV environments of northern Eurasia required major physiological adjustments. Again, Neanderthal DNA provided ready-made solutions. Several Neanderthal variants affect keratin production, the protein in skin, hair, and nails. In Ice Age Europe's cold, low UV environment, these variants likely helped by making skin more protective and possibly affecting hair thickness to improve insulation. Today, these same variants influence whether you have straight or curly hair and how quickly your skin tans. The BNC2 gene, inherited from Neanderthals, affects skin pigmentation and freckling in Europeans. The OCA2 gene variants from Neanderthals influence lighter skin, hair, and eye color. These weren't cosmetic changes. They were survival adaptations. Lighter skin in low UV environments allows more efficient vitamin D synthesis, helping prevent deficiency-related diseases. In the Arctic, some populations carry Denisovan variants affecting fat metabolism. These genes help generate heat from brown fat tissue, crucial for surviving extreme cold. Modern humans didn't evolve this capability themselves. They inherited it from people already adapted to harsh environments. The speed advantage was extraordinary. Instead of waiting for beneficial mutations to randomly appear and slowly spread through populations over thousands of generations, modern humans acquired entire biological systems instantly through reproduction. They inherited immune networks, metabolic pathways, and physiological adaptations that had already proven their worth in Eurasian environments. This genetic inheritance explains human success in colonizing virtually every terrestrial ecosystem, despite lacking specialized anatomical adaptations. We didn't evolve unique features for each environment. We incorporated pre-evolved solutions from populations that had already solved those environmental puzzles. The archaic genes that saved us represent sophisticated solutions to ancient problems, solutions that allowed our species to rapidly adapt and thrive in environments that would have otherwise taken millennia to successfully inhabit. Yet this genetic inheritance came with hidden costs. Evolution doesn't plan for the future. It only selects for what works right now. What we're witnessing is evolutionary mismatch on a massive scale, ancient solutions creating contemporary problems. The most dramatic example emerged during the COVID-19 pandemic. In 2020, scientists identified the single largest genetic risk factor for severe COVID-19. It's a cluster of genes on chromosome 3 that dramatically increases your chances of needing hospitalization or mechanical ventilation if infected with SARS-CoV-2. When researchers traced the origin of this deadly variant, they made a shocking discovery. The entire risk haplotype, a 50 kilobase stretch of DNA, was inherited intact from Neanderthals. It's carried by roughly 50% of people in South Asia and 16% of Europeans. In Bangladesh, 63% of the population carries at least one copy. This isn't a coincidence. The haplotype's high frequency in South Asian populations suggests it was under strong positive selection at some point in the past. It likely provided crucial protection against ancient pathogens that circulated through those regions for thousands of years. But when confronted with the novel coronavirus, this same genetic variant became a death sentence for many carriers. The mechanism involves immune system dysregulation. The Neanderthal haplotype contains variants affecting chemokine receptors like CCR1 and CCR5, which coordinate immune responses. In COVID-19 patients, these variants appear to trigger the kind of hyperinflammatory immune response, the cytokine storm, that destroys lung tissue and kills patients in intensive care units. A gene that protected against ancient diseases for 40,000 years suddenly became one of the pandemic's hidden killers. What evolution selected for in one pathogenic environment proved catastrophic against a different pathogen in a completely different era. The blood clotting story reveals another cruel irony. In the dangerous world of our Paleolithic ancestors, rapid blood coagulation was life-saving. A deep cut from a stone tool or animal attack could mean death from blood loss. Genes that sealed wounds quickly prevented fatal hemorrhaging and blocked pathogens from entering through damaged tissue. Several Neanderthal variants promote hypercoagulation, blood that clots faster and more aggressively. The SELP gene variant increases production of P-selectin, a protein that initiates clot formation. In ancient environments where traumatic injuries were common, this was a powerful survival advantage. But in our modern world of longer lifespans, processed foods, and sedentary lifestyles, these same genes have become cardiovascular time bombs. The variants that once prevented our ancestors from bleeding to death now increase our risk of strokes, heart attacks, deep vein thrombosis, and pulmonary embolism. What saved lives 40,000 years ago kills people today. Metabolism tells a similar story. Neanderthal variants in genes like SLC16A11 create thrifty metabolism, efficient fat storage that helped ancestors survive famines. Today, these same variants drive epidemics of type 2 diabetes and obesity in our world of constant food availability. The SLC16A11 risk variant is found in up to 50% of people with Native American ancestry. Even our brains carry the marks of this ancient bargain. Neanderthal variants show associations with depression, nicotine addiction, and altered circadian rhythms. Many of these variants cluster near genes regulating sleep-wake cycles, suggesting they were originally adaptations to different patterns of daylight and seasonality in northern latitudes. One rare Neanderthal variant significantly increases nicotine addiction risk, remarkable since tobacco was entirely absent from Paleolithic environments. The variant likely affects underlying neural reward pathways, its addiction potential only revealed when humans encountered this novel psychoactive substance tens of thousands of years later. The psychiatric connections hint at deeper neurological differences between human species. Some research suggests individuals with higher Neanderthal ancestry show altered brain connectivity patterns, with increased visual processing capabilities but decreased social cognition networks. These cognitive trade-offs may reflect fundamental differences in how Neanderthal and modern human brains were organized. This is our evolutionary inheritance, a complex genetic legacy that simultaneously enabled our species' success and created vulnerabilities we're still discovering. The ancient bargain struck through interbreeding gave us survival tools for one world, while making us vulnerable in another. This pattern of ancient advantages becoming modern curses isn't universal across our genome. The distribution of archaic DNA across modern human genomes isn't random. It's a battlefield map, showing where ancient genes won or lost their fight for survival. Some regions of our chromosomes are packed with Neanderthal and Denisovan sequences, while others remain completely devoid of archaic DNA. These patterns reveal natural selection's ongoing refinement of our hybrid genome. The most striking example is what scientists call Neanderthal deserts, vast genomic regions where archaic DNA has been systematically purged. These deserts aren't accidents of genetic drift. They're the result of powerful selective forces acting against archaic variants that proved incompatible with modern human biology. Brain development genes show the most dramatic depletion. Regions controlling cerebral cortex formation and neural connectivity remain almost entirely free of Neanderthal sequences. This suggests that Neanderthal variants affecting cognition, language, or social behavior created disadvantages severe enough to be eliminated from the gene pool within thousands of generations. The X chromosome shows particularly strong purging of Neanderthal DNA. In males, who carry only one X chromosome, any deleterious variants would be immediately exposed to selection. The near-complete absence of Neanderthal sequences on the X chromosome indicates that many archaic variants were mildly harmful, not lethal, but disadvantageous enough to be gradually eliminated. Conversely, genomic regions where archaic DNA makes up 50 to 70% of sequences tell the opposite story. These are the battlefields where archaic genes won decisively. Immune system genes show the highest enrichment, with some toll-like receptor variants reaching near fixation in certain populations. The selective advantage was so strong that these archaic variants swept through populations, replacing the original modern human versions almost entirely. Geographic patterns reveal how different environments shaped this genomic battlefield differently. The Neanderthal chromosome 3 haplotype that increases COVID-19 risk shows striking geographic variation. It reaches 63% frequency in Bangladesh, but drops to 16% in Europeans, and is nearly absent in East Asians and Africans. This distribution pattern tells a story of ancient selection. The haplotype likely provided crucial protection against pathogens that were endemic to South and Central Asia for millennia. In those environments, carrying the variant was advantageous enough to reach high frequency through positive selection. But in regions where those specific pathogens weren't present, the haplotype remained neutral, or even slightly deleterious, explaining its lower frequency elsewhere. South Asians carry more COVID-19 risk variants, not because they're more Neanderthal overall, but because their ancestors encountered specific disease environments where this archaic solution provided a survival advantage. The brain protection mystery deepens when we examine which specific neural functions remain pure in modern humans. Genes controlling language development, abstract reasoning, and social cognition show the strongest depletion of archaic DNA. Meanwhile, genes affecting basic sensory processing and motor control retain more archaic variants. This pattern suggests fundamental differences in cognitive architecture between human species. Neanderthal variants, affecting visual processing or spatial reasoning, might have been compatible or even beneficial, while variants affecting language or social cognition created disadvantages in the increasingly complex social environments that characterized modern human societies. Recent research reveals that individuals with higher Neanderthal ancestry show altered brain connectivity, enhanced visual processing, but reduced social cognition networks. These cognitive differences may have contributed to Neanderthal extinction if their brains were optimized for individual survival, while modern human brains favored social cooperation. This created a mosaic of human adaptation, where different populations carry different pieces of our archaic inheritance. Arctic populations carry Denisovan cold adaptation variants, while high-altitude groups retain different archaic variants than lowland populations. Collectively, humanity retains a diverse toolkit of ancient solutions distributed across our species. The ongoing refinement process hasn't stopped. Natural selection continues to act on archaic variants in modern environments. Some variants that were neutral for thousands of years are now under positive or negative selection due to changing disease environments, dietary shifts, or lifestyle changes. The genomic battlefield reveals that our hybrid genome isn't random, but a curated collection of ancient solutions that passed survival tests. The archaic DNA that persists today represents the best of hundreds of thousands of years of Neanderthal and Denisovan evolution, genetic variants that proved their worth across diverse and changing conditions. This genomic battlefield tells a larger story about what happened to our archaic cousins. The story of Neanderthal and Denisovan extinction needs rewriting. They didn't simply disappear, they were absorbed, becoming living ghosts within our genome. This wasn't the violent replacement scenario once imagined, but something far more complex and, in many ways, more profound. Consider the mathematics of genetic preservation. While individual modern humans carry only 1 to 4% archaic DNA, collectively we preserve much more. Researchers estimate that approximately 20% of the entire Neanderthal genome survives today, scattered across billions of people worldwide. Different individuals carry different fragments, like pieces of an ancient puzzle distributed throughout humanity. This collective preservation means Neanderthals didn't truly go extinct. Their genome still exists. It's just been disassembled and redistributed across our species. In a very real sense, we are their descendants as much as we're descendants of the African populations that gave rise to modern humans. The same principle applies to Denisovans. Despite being known from only a handful of bone fragments, their genetic legacy lives on in populations across Asia and Oceania. Some Melanesian individuals carry up to 6% Denisovan DNA, representing substantial genetic contribution from a population we barely understand archaeologically. This absorption process reveals something remarkable about human nature. When our ancestors encountered other human species, they didn't see monsters or competitors to be eliminated. They saw something familiar enough to mate with, to form relationships with, to integrate into their communities. The genetic evidence suggests these weren't isolated incidents, but part of a broader pattern of human interaction and integration. The ongoing evolutionary experiment continues today. We're still being shaped by these ancient encounters in ways we're only beginning to understand. Every generation, natural selection continues to refine the archaic DNA we carry, increasing frequencies of beneficial variants while eliminating harmful ones. Modern medicine is revealing new layers of this ancient influence. Genetic studies of disease susceptibility, drug responses, and physiological variation consistently trace back to our archaic inheritance. The COVID-19 pandemic was just the most dramatic recent example of how ancient genes continue to influence modern health outcomes. Climate change may activate other dormant aspects of our archaic inheritance. As environments shift and new diseases emerge, genetic variants that have been neutral for millennia might suddenly become advantageous or disadvantageous. Our archaic DNA represents a reservoir of genetic variation that could prove crucial for future adaptation. The absorption wasn't just genetic, it was cultural too. Archaeological evidence suggests technological convergence during the period of overlap. Neanderthals began making jewelry and sophisticated bone tools, innovations similar to those of modern humans. Were they learning from us, or were we learning from them? The boundaries blur when you realize these weren't separate species replacing each other, but related populations gradually merging into one. Every human alive today is a walking testament to those pivotal meetings 50,000 years ago. When you look in the mirror, you're seeing the outcome of countless ancient encounters between different human species who chose cooperation over conflict, integration over elimination. We carry within us the genetic memory of our species' most crucial decision, to see the humanity in others, even when those others were literally different species of human. The ghosts in our genome aren't just historical curiosities. They're active participants in our biology, influencing everything from our immune responses to our susceptibility to modern diseases. Understanding this inheritance isn't just about the past. It's about understanding ourselves and our future evolutionary trajectory as a species shaped by ancient encounters we're still discovering. This is what it means to be human in the 21st century. That we are hybrids, walking libraries of multiple human species that once shared this planet with us. Every breath you take is powered by genes refined in Neanderthal lungs. Every immune response shaped by Denisovan adaptations. Every heartbeat carrying the genetic memory of ancient encounters. These secret meetings reveal evolution's most profound truth. We never stopped evolving. The past shapes the present in ways we're only beginning to understand. If this journey through our hybrid heritage left you questioning everything you thought you knew about human evolution, don't let it end here. Subscribe to uncover more evolutionary mysteries hidden in our DNA.